And now is a much anticipated presentation by Pardis Kazemian, and who's coming to us also from University of British Columbia. Is in the Levitz lab, and she is going to talk about the CRISPR-mediated gene therapy now for GNAO1.
Hi everyone. I want to start by saying thank you to the Bell Foundation for giving me the opportunity to speak here today. So I'll be talking about developing a CRISPR-mediated gene therapy for GNAO1 encephalopathy. And we are based in Vancouver. So the top left photo is BC Children's Hospital, where our lab is located. And to the right is the Center for Brain Health at UBC, University of British Columbia. And to the bottom of that is our beautiful campus. And to the left is downtown Vancouver. Now getting into GNAO1, we've seen multiple versions of this by today, so I'll keep it short. So GNAO1 codes for G-alpha-O, which is a subunit of a heterotrimeric G protein complex, GO, and it binds G protein coupled receptors on the cell membranes. When inactivated, it is bound to GDP and G beta gamma subunits, and upon activation, it binds GTP and releases from the dimer G beta gamma, and then each of the subunits then go on later to have downstream functions. For example, G-alpha-O goes on to induce calcium channels, inhibit potassium channels, and in that it inhibits neurotransmitter release and regulation, such as dopamine, adenosine, and GABA. And then one of its most studied function, it inhibits cyclic AMP regulation. But all of that together summarizes into neurotransmitter release, neural development, synaptic plasticity and excitability, movement function, and also a role in Gerd channels, which can hint into the epileptic phenotypes. So what the mutation we're interested in is the R209H, which is classified as a movement disorder. And we actually work with a local family patient that harbors this mutation, and they've generously provided us with the iPSC line from the patient. Which brings me to my project aims. So the first aim would be to develop and optimize a CRISPR genome editing approach for targeting GNAO1 in exon 6, which is where the mutation lies. So this would be done in a relatively easy to transfect cell line like HEC293 cells to screen multiple guide RNA sequences with the DNA repair template to optimize our gene editing approach. And the best combination of that will be carried forward into AIM2, which is the in vitro and ex vivo testing of the CRISPR correction therapeutic approach in therapeutically relevant cell lines. And these cells would be iPSCs differentiated into neurons and then transfected with our CRISPR therapeutic approach. And in parallel, primary ex vivo cortical neurons generated from the R209H mice, which we will also use to transfect and look for genome editing outcomes. But ultimately, our goal is to do a preclinical testing in the GNAO1 mouse that harbor the R209H mutation. So I guess now we have a pretty good background for how CRISPR works from Dr. Levitt's talk, so maybe I'll skip through this. And delivery is also very important, and we now know about the lipid nanoparticles that we use to deliver the CRISPR components to the cells. And they are uptaken through the LDL receptor pathway. So the first thing I ever did on this project was to screen multiple guide RNAs targeting the whole GNAO1 gene across multiple exons. And this was done to test whether our delivery and therapeutic approach can even be used to target the GNAO1 gene. So I designed 6 guide RNAs targeting anywhere from exon 3, 5, and 6 to maximize our opportunity to target the gene and knock it down and be able to measure a meaningful mRNA reduction. So these are qPCR graphs from mRNA, relative GNAO1 mRNA levels, and we do see that most of the guide RNAs do have a significant reduction capacity in mRNA. And most specifically, guide RNA number 3, 5, and 6 are the most efficient ones. And in fact, when you transfect the cells with a combination of these 3 guides, you do get more knockdown as opposed to just a single guide RNA like guide RNA 3. So this multi-guide RNA system is used as our control for transfections. But the next important thing is that we want to correct the mutation. So we have to design guide RNAs that target the mutation and are in close proximity to where you want to correct the variant. So these actually all were designed to target exon 6 around a mutation with an appropriate SSODN, which is a DNA repair template. And we see from these graphs, again, on the left is relative mRNA levels of GNAO1, that guide RNA 5, which was also used in the knockdown experiments through guide RNA 9. Guide RNA 5 is the most efficient guide RNA to target the gene. And also on the middle graph is DNA sequencing. So at the DNA level, we do see that guide RNA 5 results in the most edited alleles in the pool of the DNA that was sampled. So on the right graph, which is the most important one, I do want to mention that we did get around 10% homology-directed repair from just a single administration of these guides. So from all of these graphs, guide RNA5 is our ultimate candidate for HDR correction of R209H. And then after choosing this guide, we will be testing the system in our iPSCs. I also do want to mention that we do have an active ongoing project to test a similar approach with a base editor, specifically ABE8E for targeted correction of R209H. This was kind of a low-hanging fruit because this base editor is able to correct an AT base pair back into a GC base pair through a series of chemical reactions. So it would be appropriate to use it with the guide RNA5 that we screened and showed that it has efficiency in targeting the gene, and be able to revert back the mutation back to the original base pair. And then this is done through, again, ex vivo primary cultures, cortical primary neurons from the GNAO1 R209H mice. So the next step was to assess our iPSCs and our therapeutic approach in the iPSCs, but because this was generated very recently, we had to first characterize our iPSC lines from the patients. And this is a standard procedure done every time an iPSC line is created to show that they are in fact pluripotent and are able to be induced into any different cell type that we're interested in. So typically we can look at pluripotency markers such as these four, and through staining we've shown that these cells are in fact expressing these pluripotency markers. And in a trilineage assay, when iPSCs are differentiated into the three different germ layers of endoderm, mesoderm, ectoderm, we can show that they are in fact able to differentiate into all those three different lineages alongside the healthy control line, which is the darker blue. So 2 markers from each lineage, and then we show that they are expressing those pluripotency, lineage-specific markers when they're differentiated into those lineages. And of course, we had to confirm that they do in fact carry the mutation. So through sequencing, we see that on the upper graph, the GNAO1 iPSC cells from the patient do have the A base pair, and from the healthy control line, it's a GG. So in these iPSC lines, we did a neural rosette assessment, which is a typical assay that mimics neurulation during neurodevelopment. So by measuring the lumen size, we can say something about the phenotype of how neurulation is affected given a mutation is present. So on the left, through staining of ZO1, which will target the lumen, which you can see on those circles on the embryoid body. So these are 3D spheric embryoid bodies. And on the right is one from GNAO1 patient. And while the lumen size area are not significantly different, we do clearly see a morphological phenotype. And lastly, the ultimate goal of this is to do a preclinical testing of our LNP CRISPR approach for targeting the mutation. So once all of this is done in iPSCs and also ex vivo primary cultures of neurons, all of the optimized parameters will be carried forward into intracerebral injection of the drug into the R209H mice. And these are the mice that were generated in Dr. Newbick's lab. And then we will do a short-term and long-term biochemical assays. Short-term, we'll do sequencing to see how much of the cells we're able to in fact correct. And for long-term, we will also look at the behavior phenotypes, for example, the open field that was characterized as the phenotypic difference on these mice. And with that, I want to say thank you to my first supervisor for gifting me with this precious project and also supporting me throughout. And also my lab members that have been really supportive throughout all of my experience so far. And our collaborations with the Pilati Lab, who help us with the iPSC work, and Dr. Nubik for providing us with the mouse model. And also to the Nanomedicine Innovation Network and BCCHR for providing funding for this project, and also to the patient family who generously provided us with the iPSC line. And that's my last slide.
Thank you, Pardis. Questions?
Thank you for your presentation. I was just wondering, do you see any indel formation, insertion deletions?
Yeah, so in these graphs, so actually the middle graph, these are all experiments done at the same time in parallel. So they were plated at the same time and then transfected at the same time. So the middle graph is the indels. So percent editing refers to indels. So there's about 15% indels in that guide RNA5. And then there's a 10% HDR correction when the template is present.
So aren't you afraid that you get double targeting? Because now your guide is outside of your mutation. So you can get retargeting of your, you can also get targeting of your wild type allele.
Yeah,
So
that's true. And ideally, guide RNA 7 and 9 had the mutation sequence in them. So I was really looking forward to them being the best guide RNAs. But there's also other approaches we can take, like mutating the PAM sequence and making sure that doesn't change any amino acid differences. So that's one way to create a secondary mutation that's sort of silenced, but it will inhibit the guide from rebinding and creating more indels.
Yeah. Okay.
Great presentation, thank you.
Thank
Very
you.
nice work. So one of the things that I'm a little puzzled is the efficiency of HDR in 293 cells that should, I sort of expected it to be, 50, 60, 70%.
Mm.
To what extent do you think the expression levels or lack thereof of GNAO1 in 293 cells drives this apparently low efficiency of gene editing, right? Because it's a gene that either is not expressed or expressed at a very low level. So do you think that the chromatin access actually impacts your ability to mediate the HDR?
Yeah, and that was sort of the reason why we chose an easy to transfect cell line. So we were just looking at relative efficiency of each guide compared to one another and not maybe total on their own. So while it may not be that over 50%, but one of the guide is going to perform better than the rest. And then ultimately we would choose that guide and then go into a cell line that's more relevant, like neurons that express the GNAO1 more, and then we can just optimize that guide further.
This was more just a question about the general mechanism, right?
Yeah.
Of editing genes that are expressed versus not, and to what extent, because we know that CRISPR can cut in areas that are not even expressed, right? But HDR may be something that you actually can't do if the gene somehow is not expressed in your target cell. That was just the only comment. I get it that you're just screening the guides and all of that.
Yeah, and I've done some Western blots and qPCRs initially in HEX and neuroblastoma cell lines, and they do express GNAO1 relatively to the same levels. So because of the easy, relatively easy, working conditions with HEX, we chose to just use that as our screening. So, yeah.
Hello? No. All right. Thank you very much, Pardis.
All right. Thanks for listening.